for confined out in chain. Actually, this is first time for me to meet him in person. As far as I know, he is a very famous person for the physical ecology. He's also most famous study is the experiment that slide mold single cellular organism can solve maze. Maybe today we can hear that study. Anyway, then we start now.
Thank you for your kind introduction. So, good afternoon, everyone. So, I'm Toshiyuki Nakagaki. So, today I will talk about this topic, so behavioral responses of single-cell organisms to some dilemma situations. Okay. So, this shows topics of my today's talk. So I suppose 60 minutes talk and 30 minutes discussion. And please don't hesitate to interrupt me when I'm talking. Anytime you can ask me anything. So first, so topic 0. So I will introduce our experimental organism. As the chairperson mentioned, the slimoles. The scientific species name is Physarum polycephalum. English-speaking people say Physarum polycephalum. But those
computer
speaker. The slimoles. The Physarum is one of Protists and the protozoa. And the protozoa is a conventional term, and the Protist is a current term. Okay. The Protist means single-celled eukaryotic organisms. Okay. Do you know the technical term is eukaryote? Okay, you don't know. You know. If you know, raise your hands. Okay, I see. So all of organisms can be divided into two groups: eukaryote and you know how to say this? Here's a group. Prokaryote. The eukaryote has a nucleus in a cell. Okay. A nucleus contains genetic materials of DNA. In the prokaryote, they have no nucleus. So typical prokaryote are the bacteria and the archaebacteria. The rest of organisms, all of the rest of the organisms is eukaryote. Okay, and the animals and the plant and the fungi and the mushrooms and the slime. Okay. So Protist is a single-celled eukaryotic organisms. So I have studied some behaviors of this Protist for many years, over 30 years. So this shows the experimental organism, kind of paramecium, and so on. So today I will talk about this organism, slimoles. These are kind of amoeboid organisms. The very big amoeba like this, still small, okay? But sometimes they grow. They can grow up to square meters, so quite huge amoebas. Okay. So because this is big enough for us to recognize by naked eye, so then this is very nice thing for the experiment. So we can feel something when we culture this organism day by day. Okay? And anyway, so this is the slimoles. So I will talk about two topics on the behaviors. The first one is multiple causes of action at delicate situations. The second one is foraging behavior in multiple distributed food pellets. And then lastly, I will summarize my talk. You mentioned that diversity or flexibility of behavior in the same or similar situations. Okay. Let us begin with the brief introduction of the organism. So I will show you the very nice movie originally made by German guys, the Karl-Heinz Bergman, almost 20 years ago, over 20 years ago. The title is very, very lovely, like nothing else. Okay, total playing time is almost 1 hour, 43 minutes. I will show you very short clips of this movie. Okay.
The slime mold moves across the ground, though it is less dramatic than the time-lapse camera makes it seem. Every centimeter takes the slime mold a full hour. When it seems to pulsate violently, it's only because after each advance, it pulls back a bit. Slime molds need moisture. As soon as it rains, they stream out of cracks and crannies and set off on their journey. They flow over algae and bacteria, and if they come across a large tree fungus, all the better. It too will be dissolved and digested. As if by magic, they form veins to spread the nourishment around their body. So far, nobody knows what makes these tubes appear and disappear.
So, but I know.
When two of them meet, they flow into one another and move on as a single larger organism. It's as simple as that. In dry conditions, everything changes. Then they must get back as quickly as possible into the damp soil. Or else they must seek salvation in one last quick change act.
Okay, and so I will say a few words on the basic biology, cell biology of plasmodium. Okay, so you saw the rhythmic contractions in the movie. The mechanical force is produced by the actin-myosin filament. These are muscle proteins. Okay, there are such proteins in the cell. Okay, very similar to that last. Okay. And the rhythmicity of the contraction comes from the biochemical oscillations, like so calcium ion and ATP, etc. These kinds of biochemicals can oscillate. Okay, so that this is the source of the rhythmicity of the rhythmic mechanical contractions. So, according to this rhythmic contractions, you can see the shuttleless flow of the protoplasm all over the cell. Okay. So this is a typical schematic illustration of the shape of the extending organism. So this moves so this way. Okay, this is the front sheet-like structures, and intricate network of veins develops toward the layer like this. Okay. In the sheet-like structure at the front, the internal structure looks like porous media like this. So you can see the thick bundle of the actin-myosin filament like this. So then in the vein, there are two layers. Outer cortex is gel, and the inner part is azole. If this part contracts and the rear part relaxes, the hydrostatic pressure is produced between two parts. And according to this internal pressure, the flows backward. And at the next phase, the opposite phase, the frontal part relaxes and the rear part contracts. The flow direction is switched. So then we can see the shuttle streaming. Okay. So this is a cell biology of the plasmodium. And this shows
Make sure so this organism is not composed by membrane, by layer membrane, but it's kind of gel or something.
yeah, but the most outer part of this cortex, there is also a biomembrane. Okay. Yes. All of this amoeboid body is wrapped by the biomembrane. Okay. But of course, you can see some complicated structures, the kind of invagination. This is a space come into some layers of gel kind of thing. Okay. なんか、
まだなんかエコー。
そうですね。
なんですかね。
なんでですかね。
ラップトップのスピーカー。ベヒーミューティンのこれはスピーカーじゃないですか。じゃない、やっぱりここから聞こえてますね。
そうですね。
そういうの問題だ。
消えない時が。なるほど。
でも、今と音といいですか？
いいです。もう音がいいです。はい。それですね。なんか気になりますね。
そうですね。ちょっとそれです。
はい。では一回も。はい。すみません。
すいません。
いい。ありがとうございます。それで、これ。はい。And this is the life cycle of this organism. So what you saw is the plasmodium. So this stage of this life cycle, this is a vegetative stage of the organism. Okay. This is a huge amoeba, but this amoeba is a multinuclear organism. Okay, so now we come to the first topic, so multiple causes of action, a delicate situation. Okay, so this topic is based on these references, 3 references. I like to mention 3 key contributors. So Nakagaki Seiji-san has done the experiment. And Ueda Keiichi-san and Nishiura Yasumasa-san have done the mathematical modeling and mathematical analysis of the model. Okay. So this experimental setup is quite simple. This is a top view of the experimental arena, so narrow range. So the thymos is initially inoculated at this left end of narrow range. So then they start to move and they extend. And here we have the toxic zone. Okay. So later the organism come to this toxic zone. Okay. So then in this space, there's no food, of course, and no free space. And the space is polluted by the waste material released from the organism itself. Okay. So the organism is confined in this space. So that in these situations, I may say to go or not to go. So that is a question when encountering a toxic zones. So then I think this is kind of dilemma situations. Do you agree? Okay. So this is the result. You can see the 3 different type of actions. So this is penetrations and the rebound and the return and the splitting. You can see the 2 extending front here this way and there that way. So, but they are still connected. Okay, so slime moles tend to penetrate at low concentration of the toxin and the quinine, but tend to rebound as the concentration increases, 8 millimolar. The splitting is also possible here. And the interesting thing is that the 3 different actions are possible at the same condition, same concentration. For example, the 6 millimolar from experiment to experiment, so we can observe one of them. Okay, so the percentage of 3 different actions, so depends on the concentration. Okay, so this is experimental result, and this slide shows. Sorry. The result and the profile of cell thickness. Okay, so this the same result but a different expression in the presentation of the result. So this is a space in the one-dimensional space along the narrow lane. Okay, so this axis is the time. And the z-axis indicate the thickness of the film. Okay, so here you can see the thick front and move this way at almost constant speed, so almost nearly straight lines. Okay, so then they come to the toxic zone here, so around 7 centimeters. They stop moving like this. So when come to the poison, the slime will stop for a long time. So like this. So once start to move like this, it keeps moving first like this. Okay. And this, so waiting time varies so greatly from case to case. So very short and very long. We cannot predict. So depending on the experiment. Okay. The thick front disappear. So as indicated by red arrows, the thick front disappear once just after encountering the poison like this, and recover like this later. As indicated by green arrows, a leading front appears in the middle of the back, then going backward like this. And please see the yellow, the arrow. The front sometimes moves a little around the keen zone like this. The fluctuate at the position of the thick front. So these features are to be followed by mathematical modeling. Okay, so now we try to make a mathematical model to reproduce these behaviors. Okay. Okay, so now next several minutes I will talk about model. But what I'd like to tell you is a basic idea of mathematical modeling. So please don't care about the technical details of the mathematical model. In detail of a mathematical model is not justified. Okay, so but I like to explain the basic idea of frameworks to describe the motion of the slime moles. Okay, this shows a schematic illustration, the cross section of the organism extending rightward like this. So there are 2 layers, gel layers and the zone layers. Okay, and this is enlarged picture that so zone flows. Okay, so this is a locomotion of the organism by the pressure of the contraction of the gel. Okay, so gel is elastic at a shorter time scale, and plastic in a longer time scale. So then we try to describe the elastoplastic deformation of organism. And gel has active rhythmic contraction. Okay. And this elasticity can be regulated by the biochemical regulation or kinetics. Okay, and so this biochemical regulation, the reaction, is modulated by the existence of the toxin or poison. Okay, and this gel stiffness is also affected by the flow of the zol. So because the zol flows and accumulate at some place, so gel must be expanded and swollen. Okay, so then so there are some stretch activated calcium channels at the cell. So then these channels open and the calcium ion so come into the cell. So then this is a strong modulator of the regulation kinetics, chemical reaction. Okay. So then this shows the formulation of the model equations. So the S, you can see S, the variable S, is a function of space X and time T. The subscript T means the time derivative of function S. Okay. So this equation is a physical equation of mass conservation. So this is a kind of flux, the flux of divergence of the flux kind of things. So then this is, okay, so P is a pressure. This term is a conversion between the zol and the gel. Okay, and so we also describe the motion of the gel density W like this. So you can see the conversion term like this. So the pressure is a point. Okay, the pressure is given by these equations. So and this depends on, this include the active contractions. So I don't talk about the details. And the SB is a key variable that express the active contraction. And of course, we have the dynamics of the plastic deformation of the gel at the slower time scales. And here we have the stiffness constant of the elastic force, so beta. So beta depends on some chemicals nu. And here we have dynamics of the U and the nu. So this is a biochemical reactions that regulate the stiffness of the gel. Okay, so kind of things, I don't talk about the technical details. Okay. And this shows a typical profile of some variables. Okay, this is a one-dimensional space and the X. And the U and the V, so here U and V, the biochemical reactions like this. The U is a kind of V, so nu, U, V, V. Some concentration of the zolation factors. Okay, so this is a leading front and the soft front. So the zolation factor is high, and the U is a kind of concentration is related to some concentration of the atomizing filament or kind of things, and so stiffness or something like that. Okay, and the S and the W, and this is a body. Okay. And the thick front kind of things. And here we have the kinine zone, the toxic zones. At these zones, the reaction constant of this regulation biochemical reaction is different, okay, like this. So then this is a kind of basic framework of our mathematical. So then this shows the simulation results and the comparison with the experiment and the model simulations. So, so the model can reproduce 3 different type of actions. Okay, so the penetration and splitting and the rebound like this. So then, as indicated by some colored arrows, we can see some similar type of behavior in the model simulations, but not perfect. But still, we believe we can reproduce some part of the observed phenomena. Okay. And so 3 patterns are reproduced by the slight change in the bump height. The bump is a toxic zone. Okay. So just a 10% difference or something like that. So next, I'd like to simplify this, the complicated model. So by just only 2 variables. I don't talk about the details. So anyway, so now we have 2 variables only. So U and V and kind of thing. So then this is a 1-dimensional space. The situation is similar to the previous one. Okay. And variable V is higher at the front, and U is lower at the front. Okay, so V is a kind of isolation factors, and U is a kind of stiffness or something like that. So this this part can migrate this way. So then you can see the traveling pulse or traveling waves of this part rightward. So then this is a migration of the Slimers. And here, the toxic zone. Okay, at the toxic zone, the kinetic parameter is different like this. So then this is the model equations like this. And then this is biochemical nonlinear reaction terms. And so this is diffusion terms. Okay. So then this is a model simulation. And the trajectory of the same initial state, the action of the organism, so goes to a different state due to a very tiny difference in the parameter of the kinin concentration, the height of the bump, kind of things. The penetration and the splitting and the rebound. And you can see some quiescent stage, so stop moving, so Slimers stop moving. Okay. So then this, okay, the simplified model, so we can analyze, we can see the mathematical structures of the bifurcation of the systems. So then based on this mathematical analysis, we like to a similar type of dynamics, like this, so, and a story or metaphorical story of the dynamics. So here we have the 1-dimensional array of matches like this. Okay, so this is a space, and so match is firing here. Okay, so the firing is a kind of excitable systems that is observed in the model for the Slimers. Okay, autocatalytic and nonlinear reactions. Okay, so this is so firing. So right one so will fire, but left one will not, because this is exhausted, so the burned out. Okay, and new match is so supplied at the front here. So burned out match regenerate after a while like this, and fire spreading goes on this direction. Okay, So then suppose there is a humid zone like this indicated by the red dotted box. So it will take some time until firing in this match. And the band at match will regenerate, okay, kind of thing. So then the fire is able to spread to one or two directions, okay. So it may depend on the delicate balance of humidity and the match generations, like this. So like this, we may observe and understand the multiple causes of the actions, the similar situations. Okay, so this shows delicate balance of the penetration and the rebound by different explanations. So kind of the balls moving on the landscape like this. Okay, so this ball goes this way, and the motion of the ball is modulated by the perturbation at the top. Okay, then the motion is slowing down around the top. Okay, so this corresponds to the quiescent stage, and depending on the slight difference of the initial positions and the height of this mound, so final action may be switched this way and that way or something. Okay. So then here, so I'd like to read a bit more on the subtype of the behavior. Okay. So this is a space and time plot. You can see the grayscale. So grayscale indicates the thickness of the organism. So this is a space and this is a time. Okay. Then here we have a toxic zone. Okay. So the organism come to this toxic zone and they stop moving at the first interface. Okay. So still we can see the second interface like this. In this case, this organism stopped second interface, not first interface. Okay. And in this case, the organism stopped 2 times at the first interface and the second interface. And in this case, it stopped at the middle on the 2 interfaces. So you can see this kind of behaviors. Okay. So this is some examples of the behavior, the subtype of behaviors in the penetrating action. Okay. So, and you can see some interesting and so strange behaviors. So come across over this toxic zone many times and back and forth kind of things. Okay, so we tested almost 500 repeats of the experiment. So roughly speaking, so we can classify the 15 subtypes of the, at least 15 subtypes of the action. So you can see the rich variety of action in the slime molds. Okay, so this shows the statistics of the subtypes where slime molds stop. This is a first interface and the second interface and the middle. So then the 3 possibilities. So indicated the plus and minus. So then this shows some examples. This black line indicates the frontal tip, the position of the frontal tip. Okay, this is a space and time plot. This scale bar indicates 1 hour. So then they stop at the first interface and middle and the second interface and 2 times and 2 times middle and second and kind of things. So in this case, so this is a one hour. In this case, slime does not stop at all at the interfaces. So then it's a frequent statistical occurrence like this. Okay, so then our simplified model is a kind of reaction diffusion equations with two variables. But Nishiura and Ueda studied the little bit different reaction diffusion type equations. The behavior of the reaction diffusion type equations is three variables. This is a little bit far from the biological activities, but we can so find some connection or similarities in some sense. So anyway, by this model and the equations, you can see the rich patterns of the pulse motion. So like this, and a stop at the first interface, and or the second interface, or the penetration and the rebound, and the kind of rhythmic back and forth motion or something. So these kind of rich patterns can be reproduced by the simple model equations. This is what I'd like to stress. Okay, so then I'd like to say some remarks on the multiple course of action. We observe the multiple actions emerged naturally from the dynamics. So dynamic equations, the slight difference of local reaction to a toxic zone at the front. So then this slight difference result in the difference of the global behavior of the organism. The quiescent stage so existed before the action choosing, kind of, I may say, the decision making. And the length of the quiescent stage was not predictable. Okay. The motion at the individual level can be unpredictable from local reaction to environmental cues. And unexpected transformation so intervenes between the local reaction and the individual behavior. So this transformation is a source of the behavioral diversity and the variability. So mechanism of transformation may tell us unexpected heuristics. So that is from simple local response to the behavioral flexibility. This is a message. Okay. So to go or not to go, so that is a question. The last paragraph in our papers, physical review, we described this comparison, the Slimer's behavior and kind of human behavior observed in the Hamlet. So I like to skip to read this. Anyway. Okay, so then next I'd like to talk about second topic, the foraging behavior in the multiple distributed food pellets. So slime was here, so we can present one or two or many food pellet here and there in a space. So then we may put some obstacles. So then try to see what's happened in the behavior of the slimers. This is a maze, the behavior in a maze. Okay. The many obstacles and walls. And at the initial condition, the organism spreads free over the available space in the maze, like this. Then we presented two food locations and the food pellets here and there. And almost 4 hours later, the body shape is totally changed like this. So then the part of the maze, so here and here and here, and so disappear. So these parts move onto the food pellet to absorb the nutrient as fast as possible and as much as possible. And on the other hand, we can see the thick veins that connect to food pellets. And 4 hours later from this, you can see this shape. In this case, the one thick tube connects the 2 food pellets. And this distance is almost the shortest. Okay? I'd like to mention the length of the root a little bit. So this is okay. So if we go along this lines, so black lines like this, it turns at the right angles here, like this. So then there are 2 possibilities from here to here, A1 and A2. The 2 course, 2 paths has the same distance, okay? In this case. Okay? But is the trajectory traces like this, so out in out trajectory. So the lower path is 20% shorter than that of the upper one, like this. So for this site, okay, there are 2 possibilities. Okay, the beta 1 and the beta 2 are comparable, nearly equal the distance. Okay, so let's see the thymos. Of course, you can see some fluctuations, so not the straight lines in a mathematical sense, but we can recognize the out and out so turning like this. So then this path is almost the shortest that we can see. So this showed a statistical result of the final shape of the organism in the maze. There are 2 sites, the 2 options here. This is alpha 1, alpha 2, and another 2 options here, beta 1, the beta 2. The number indicates the number of experiments and the number of the result. Okay, so for this part, these 2 options, alpha 2 is always selected like this. For beta site, okay, beta 1 and beta 2 are comparable, okay, because this is nearly equal. Okay. And I like to mention that this case. So, and in this case, the 2 paths also remains. Okay, 2 paths remains. This means a multiple connection route between 2 food patients. And here is a none, means the organisms separate into 2 subsystems. Okay. Oh, yeah. In the maze is amazing, is my joke. Okay. I like to say a little more on the various solutions. So in the previous experiment, we repeat the 19 experiment, and almost 50% nearly shortest path is obtained. And it shows some the patterns of the multiple connections like this, almost 30% of trials. This shows the separation into subsystems to individuals. That is 10% of the occurrence. Okay. So even in the same experimental conditions, so you can see the multiple behaviors, multiple patterns of the behaviors. So, and on this type of experiment, I like to mention the many factors that strongly affect the final shape of the organism in a maze. Okay. So in this case, the size of the organism, the mass of the organism is larger than that of the previous experiment. So you can see some aggregation of the body, so like this. So sometimes this aggregated part come across over this wall of the maze. So then they establish the connections through the wall of the maze, like this, indicated by red arrows. Okay, so then finally they can select. So after the presentation of the two food locations, shorter connection path than the shortest connection path. Okay? So we can point out many factors. Of course, one important factor is body mass, the size of body mass, and the amount of food pellet presented. Okay, and the atmospheric temperature and the humidity and the light illumination, kind of things. And depending on these factors, the final shape is different. Okay, totally different. And suppose the wild conditions and environmental conditions in the wild habitat. So there are many inhomogeneous distribution of the many factors, even the size and the chemical composition of the food pellet or something. So this is totally different, and the nitrogen source and the carbon source and the kind of things. So in these complicated situations, so we expect this organism can form the nice network shape of the body. Okay? But how smart they are, so it is difficult for us to evaluate and elucidate. Okay? So then, yes. Still we have the possibility of the future study on the slime moth. Yeah, please.
What would be the base wall made of? Is it higher wall or
Just a flat wall. Yeah, just we put the plastic film on the agar gels. Okay, no height, the same height, and
it's possible to?
yeah, not so difficult to come crossover. Right. Okay, then I will show you a small movie on this.
1,000 kilometers to the north is the University of Sapporo. In the Electronics Institute, a unique experiment is going on. An intelligence test for slime molds. The subject is polycephalum, in English, the one with many heads. Just how much it has in those heads we shall see. A maze is set up with two marked points, A and B. The challenge is to find the shortest route between A and B. An easy task for us. But for a slime mold, Toshiyuki Nakagaki brings his protege to the trial. Physarum polycephalum, a single huge cell. First of all, it has to be put into the maze, and that is amazingly easy. He breaks off one bit after another and moves them in. No multicellular creature could survive this treatment, but slime molds follow different rules. Nakagaki's patchwork approach is good enough. The slime mold will sort itself out. The bits of polycephalum join themselves together again and fill the whole maze. Now the actual experiment can begin to find the shortest route from A to B. He supplies food at both points. Porridge oats, Polycephalem's favorite food. The slime mold can't resist it and tucks in at once. The time-lapse camera reveals its greed. In a great wave of gluttony, it throws itself on the oats. But in spite of its haste, it follows the most direct route between the two food sources, laying down thick veins through the maze. It seems to avoid many possibilities. It pulls its veins back out of blind alleys, for example, leaving no trace behind. Also, detours are soon abandoned. And after a few hours, there is just one main vein, one clear route between A and B. Without doubt, the shortest route. The intelligence test is passed with flying colors. Meanwhile, Polycephalum, fully fed, has left the maze and is considering producing young. Not a bad idea. Slime molds are quick change artists and intelligent with it. And that's not all.
So this is an experiment, and I was young. Okay. The next, I'd like to talk about the mathematical model for this observed behavior. And the maze solving experiment, the key thing is the morphogenesis of the tubular structures of the body. So then we found that from the experiment, there's a so interesting adaptability of the organism. So current reinforcement rules. So the tube can change its thickness. So according to the flow through the tube itself. Okay? So here we have the slime mold tubes and vein. If the flow through this tube is big enough like this, so then thickness increase a little bit. So then the resistivity of the flow decrease. So the more flow is likely to occur. So this is a kind of positive feedback process. And on the other hand, the flow is not large enough. The thickness decrease a little bit. The resistivity of the flowing increase. And less flow. And the thinning and less flow, and so on. They finally died out. So this is a current reinforcement rule, and local adaptability of the tube thickness through the flow through the tube itself. So based on this current reinforcement rules, we propose a toy model. Okay. And the slime mold in the maze is modeled by the network of water pipelines like this. And water inlet and outlet are set at the food locations like this. Okay, so please imagine there is a food pellet, and the protoplasmic mass that flows back and forth between two food locations. Okay, so such a situation is modeled by these things. Okay. So the first we try to calculate the current for each types like this. So we can calculate according to the physical law of the hydrodynamics or kind of things. And based on this physical nature of the flow, next we try to describe the growth of the pipe by current reinforcement rules. So we can specify the flow rate for each local pipe. So this is physics, not biology. Biological thing is this side, current reinforcement dynamics. So then we can simulate the dynamics of the thickness of the water pipelines. So then this showed how to calculate the current. So suppose this is one example of the water pipelines that consist of the joints and pipes. Okay, this joint number I and J. Between joints IJ, so here we have the pipes IJ like this. This pipe IJ has the length and the radius and the flow rate, the three physical quantity. The flow rate is given by this hydrodynamic approximation, quasi flow. P is the pressure at the joint. The pressure difference is the motive force of the streaming. Then the conductivity is inversely proportional to the length of the pipes and proportional to the horsepower of the radius like this. So anyway, so we can then we have the water inlet here. Volume Q0 flows in, and the same volume Q0 flows out from the other food locations, so outlet. Okay, so then we can think about the volume conservation at any joint. In this, please look at joint I. There's the four possibilities of the pipes and the flow in and the flow out. When we sum up all of flow, it must be zero. So this is a volume conservation. So due to this volume conservation, we can calculate flow Qij. So this is a physics. And like this. And
You have 5 minutes left, but of course you can go ahead, but I just like to notice you.
5 minutes. So, okay. Okay. じゃあ、はい。5 more minutes, I may continue to talk. Okay. Thank you for the reminding the time limit. Okay. And this shows the dynamics of the growth of the pipe by current reinforcement rules. Okay. So this is a conductivity. So here, conductivity dynamics is given by this simple equation. In this case, a current recurrence equations. So then now we are at time t. So right-hand side of this equation, and left-hand side is near future time plus dt, and the thickness of the pipe is given. Okay. And so this is dynamics of the modulation of the thickness. This is a thickening process. Okay, by current reinforcement rules, the Q is a flow rate and absolute value. So we don't care about the direction of the flow. The F is so-called current reinforcement function. This is an increasing function of flow rate Q. And the concrete shape of this current reinforcement function is not known from the experiment. So we assume that for the sake of simplicity, power functions in the model simulation later. And so this is the current reinforcement terms. And we have another antagonistic term, the thinning process is first order kinetics. I don't talk about the detail of this model. Anyway, so according to this simple equation, so we can calculate, we can simulate the shape of the network. Okay, this is, so then the model simulations. So then, sorry, I have to mention. So here, now we have this current reinforcement functions. Mu is a model parameter exponent. Mu. The mu is 1 and the alpha is 1. So then this shows in the model simulation. And shortest connection path is obtained. But in this simulation, the length of this path is 20% shorter than that of the other. So, so then to be more realistic, so each corridor of the maze is represented by the fine meshwork of water pipelines like this. So then using this setup of the simulations, we can reproduce out-in-out turning of the pipes like this. Of course, this simulation is done in the mu is 1 and alpha is 1. Anyway, so we can reproduce the shortest path finding by slime models according to the current reinforcement model. Okay, so but this model can reproduce the multiple patterns of the network shape depending on this model parameter mu. The mu was 1. The shorter one is always selected for this simplest situation. There is one short connection and one long connections. In this situation, we can solve this model by hand. So then we can recognize the mathematical structures of the bifurcations, but I don't talk about the detail. Okay, mu is less than 1. The both paths remains, but the final thickness of the tube is thicker in the shorter. Okay, and the mu is larger than 1. Depending on the initial thickness of the pipe, one of them is selected. Okay? If the longer one is very thick at the initial conditions, so the longer one may be selected, okay, kind of things. So what I'd like to say is that so we can, this model can reproduce multiple shape of the network. Okay. So this case, you can see the many, almost 40 food pellets in a space. At the front tip, so that we inoculate the organism here, then they start to spread out toward the periphery. So this is a frontal tip and the thick and soft front. There are many capillaries. The very limited number of capillary can survive and become thicker to connect the food locations like this. It takes about 1 or 2 days to finish this experiment. And so then the location of this food pellet is the same as the location pattern of the major city in the Tokyo area. So then we can compare the functionality of the shape of the network, the railway or transportation in the Tokyo region, slime holes in the pellets. So then, okay, we introduced 3 different measures to evaluate the functionality of the network between major cities and the food pellet. The first measure is the short total length of the network. This is a kind of measure of the cost. I talked about this only in the previous slide. Now we like to focus on the 2 more things. The fault tolerance of global connectivity against an accidental disconnection. So suppose the accidental disconnection may happen here. So this guy is isolated and separated from the main body of the network. But for this network, look like circles against accidental disconnection here, but still connected, okay, through the another path kind of things. This is kind of measure of fault tolerance or robustness. The last one is a short connection distance between 2 food sources, a measure of the efficiency. So this is the best efficiency because any pair of 2 food location is connected through the direct or straight lines. But the total length is very long. Okay, so important thing is that we must observe trade-off between total length and the other 2. So this is kind of compromised product. And all of three measures are not the best, but not so bad. So using these three measures, we can evaluate the functionality, the multifunctionality of the shape of the network. This is the total length and the cost. The lower is better. This is the higher is better, the fault tolerance. And global connectivity against accidental disconnection. Okay, you can see many black balls like this. Each ball is one data obtained from the experiment. So the data distributed in this range like this. So tendency of this distribution is approximated by these red lines. So red line is obtained by the mathematical model. So I will talk about next slide. Anyway, so then you can see multiple diverse compromised product for each shape of the network like this. So that was good compromise. Sorry. Okay, so then Tokyo Railway network is as good as the Slimos network. So I think this is a very good news. And so in this model, so we have some model parameters. C is related to the shape of the current reinforcement functions. Of course, the network shape strongly depends on the current reinforcement functions. And the Q0 is water, volume of water coming from the inlet. So this is another interesting model parameters. Q0 increase like this, the networks becomes more robust and more efficient like this. So Q is closely related to the population region in transportation network. In the Slimos case, Q0 is a body mass, the size of the body. Like this. So then this dependency is quite reasonable. The larger organism and much population, this kind of network is preferred, even pay the much cost. Okay. Kind of things. Okay, so then now I like to, sorry, 2 or 3 more minutes I will finish. This shows the lesson from the foraging behavior of Slimos. The multifunctional network of body shape that reaches many food pellets is established, and it is diverse. The body shape is shaped by the dynamics based on the local responses. And the first factor is biochemical changes at the food pellets. And the second one is the current reinforcement rule of veins. And some parameters of dynamics brings about a different shape of network. So the diversity or flexibility of behaviors emerges from the mathematical nature of dynamics. So that is a bifurcation of the solution by slight difference of parameters and the initial states. So as this gap between local response and the global behavior is so big and wide, it is difficult to see and recognize. That we may be unable to recognize how they connect each other without mathematical modeling. And mathematical modeling is quite helpful for us to see these connections. And okay, this is the last slide. I summarize the topic 1 and topic 2. Multiple actions encountering the delicate toxin. So this is topic 1. Local biochemical responses to toxin so causes switching of behavior through the dynamics of model equations. the topic 2, diverse compromise of foraging behavior when encountering the many distributed food pellets, the local biochemical responses to food pellets. So bring about a variety of behavior through the dynamics of model equations. Mathematical nature of dynamical equation tells us how such behavioral diversity emerges when encountering the similar situations. So then this question, not the concluding remarks, kind of. So this invisible gap, so this wide gap is invisible, is quite difficult to see between local response and the global behavior. Is so what we may feel the free will, so behind the visible behavior. So I think this is kind of open question. And okay, I think that's all my talk. And the last two slides, just kind of message from the Max Minakata, who studied the many aspects of slime moles, quite famous person for the study of slime moles almost 100 years ago in Japan. Okay, thank you for your time.
Thank you very much for the amazing talk. So any questions?
Just want to begin with some clarification questions about the nature of slime molds. So when they learn how they spread themselves and get food or get over poison stuff, is that the only way they can move? When they move, they need to expand their body. So can they not move their whole body like bacteria, moving in there? So that reminds me more of plants rather than animals. Like plants, when they want to do how big is, they grow, and it looks like that. But the difference is that they can shrink. Is that the only way they can move?
And so, for example, this type of experiment. So, okay, and please. Okay. So, in the experiment,
1,000 kilometers to the north is the University of huge cell. First of all, it has to be.
So typically, so we culture this organism in these large containers. So then we cut some portion of the organism. So then put the new arena, so kind of things. So thinking about the natural situation of the organism, this is quite artificial conditions in a very small space, the thick amount of the organism. So then in these situations, they try to expand out first. So then once they reach to the thickness, the thin enough to the natural conditions, so they move. So this is not to expand all directions. They select one direction and to move kind of things. So then, and of course, in this culture, the open air cultures, on the agar gels, so there are many bacteria here and there. So then sometimes bacteria and fungi, kind of many microorganisms are there. So depending on the composition of this microorganism, they try to choose this way and that way and the kind of things. And of course, we put the food pellet oats flakes here. So this is a kind of favorite food for them. So they try to gather but we always give the oats flakes, oats flake again, oats flake again. So the nitrogen will be deficient. So then at that time, we present a kind of nitrogen source, like milk, nut, kind of. So then they like much nitrogen source than the oats flakes kind of things. But of course, they like to move and
Thank you.
Any other question or comment?
Hello, thank you so much for your talk. That was awesome. So I just have a quick clarification question. Kind of glad you brought this up because I wanted to know, like, for your topic one, the experiment you did, it said you did 480 repeats. And I was wondering if, how many different cultures those repeats came from. So if if you were using the same culture for all those repeats, or if you varied between cultures for those.
Culture means the, of course, in the experiment we culture the large one. Yeah. So then we cut out a small portion of the organism from the same and identical large organisms. So then according to this procedure of the experiment, we can prepare the similar initial conditions of the experiment, but up to 20 or 30 repeats only. So then next day, so we try to use a different organism, so kind of things. So of course, strictly speaking, it is in principle difficult to prepare exactly the same initial conditions of the experiment. So we have to take care of some difference of the initial condition of the experiment. Of course, the internal state of may be different from case by case. So in principle, it is, I think, difficult, impossible. Yeah. But what we can do is a decrease of the initial difference. So try to be similar, try to be homogeneous. But so yeah, that's a point. Right?
So then I was kind of wondering, if you're using each culture, let's say 20 to 30 iterations of the experiment. So then let's say for 480, I can't do quick now, but 20 something. If you're going over 20, using maybe up to 20 different organisms to run the experiment, I was just wondering if you noticed any similarities between those organisms versus other ones.
So kind of the individuality.
Yeah.
Kind of. Yes, talking about genetic background, we can prepare the same genetic background, kind of. So then, but the state of the internal state, some set of the biochemical concentration. Of course, there are many kinds of biochemicals inside the cell. The set of the concentration is, of course, different from the state and the time, or even in the same individuals, this part and that part, the chemical state is different. So of course we can cut out this portion, that portion. So looks similar, but of course, we can observe the tendency of the, action, the tendency to select the, yeah, so we difficult to, so we can also difficult to recognize individual kind of individuality of the organism.
Individuality, I feel that.
So individuality means that the difference of the, yeah.
In that sense, it may be important if the slime mold can modify or change the behavior in terms of the experience. In other words, to slime mold learn from experience.
Yeah, I think that's true. What we want to do is even in the almost a similar initial state of the organism, so resulting behavior is totally different. So we like to show this possibility. So of course, then if the initial state is different, so of course the resulting behavior is different, of course.
Cool. Thank you. Other question, comment?
Thank you for the presentation. My question is about the first experiment using Protists. And so I wonder, the slime mold seem to react or respond before reaching the toxic zone.
Oh,
There.
Kinine, so, the slime molds touch to the toxic zone. So then they can recognize, so this is a poison or something. It's a touching sense. Okay. Otherwise, they can not recognize the existence of the poison. But if the poison can, smelling substance like ethanol or vanilla, so they can sense this chemicals. So far from the source of the chemicals, so they can sense and they try to avoid and they try to come closer to this so kind of things. But the kinine is not a smell, and 答えてますか? My answer can meet your question. And the oat flake so does not smell, So then the slime molds go through like this. So then they can not recognize here is just oat flakes and go through these kind of things. Once they touch, so they can find.
Thank you for your presentation. And this question can relate with the argument of the learning capacity. And does slime mold has a capacity to transporting information each other, or just following static mechanism? Does slime mold does they have any system for transporting information?
So the question is, what is the information in the slime molds? Of course, and So when they touch the poison or oats flakes, of course, they absorb some chemicals from this sort of object. So then such a chemical can flow over the body, kind of things. But of course, this is one of the information. But so important information is a kind of, some local response. And changing the stiffness of the body. So the ceramics touched to the poison, they become stiff at the local part. And food, they become the soft, like this kind of things. And this kind of change in the local change in stiffness of the body. So then this can result in the different global pattern of the body. So then what does this mean? Information is a kind of that's all I have to say. Information is not concentrated at space and time, not concentrated in space and time, so distributed. Okay, so depending on the dynamics or kind of pattern formation dynamics, this
the motivation of this question is because when I think about the consciousness or the neural network, I remind about Simon's phenomenon, and the mechanism of Simon is simple, and it has a state that binary state like state, and it is really similar with neuron, but neuron has a clearly information, information, but natural mode is difficult to clearly they have information. So
okay.
What is this different?
You said the information in the neural network is clear. So what is the information in the neural network? I'd like to ask. Just a kind of pulse.
Yeah.
So, but I think the timing of the pulse is important, and some I think, okay, so we have some levels of the observation. Of course, we can observe the pulse sequence of the neuron. And so suppose that we have some kind of information like terms, okay, for example, of decay. So there is some big gap between the concept of the term of the Tokei and the pulse of the neural network. So how do you say what is the information about this? Of course, in the Schleimholz, something like pulse of the neuron. So we can easy to find some reaction, the kind of physical reaction in the Schleimholz. Of course.
Yeah, thank you. It's really difficult. But I think I think the function of memory is really important, but I think Simon has also having memory because they have chemical state or something.
Yes, in the different type of experiment, so we can find the capacity of the memory, and so memory of time sequence of the event, or memory of the shape of the arena, or kind of things. But how? Okay.
Time for the question. Okay, again, thank you very much for wonderful talk.
You kind of mentioned that it is quite difficult to express the what I'd like to say in English, and quite kind of